Hello everyone, welcome to my channel Dr. Tapati's presentation. Myself Dr. Tapati Vanzade. Today topic of the presentation is isoenzymes or isozymes. I hope this video will be helpful for you.
Recently, I have started discussion on enzyme regulation. Already, I have discussed about allosteric regulation, activation of latent enzymes, compartmentation, and regulation of enzyme quantity by enzyme synthesis and enzyme degradation. You should watch those videos. Today, in this video, I am going to discuss about isoenzymes. This is very important topic. Isoenzymes are the multiple forms of an enzyme catalyzing the same reaction. If you consider this picture, these are three forms of an enzyme. They are catalyzing same reaction. That means they are acting on same substrate, producing same product. But they differ in their physical and chemical properties, including the structure, electrophoretic and immunological properties, Km and Vmax values, that means kinetic properties, pH optimum, relative susceptibility to inhibitors and degree of denaturation. There are many possible reasons to explain the presence of isoenzymes in the living systems. Like isoenzymes are synthesized from different genes. As for example, malate dehydrogenase of cytosol is different from that found in mitochondria. Oligomeric enzymes consist of more than one type of subunits, as for example, lactate dehydrogenase and creatine phosphokinase. An enzyme may be active as monomer or oligomer, as for example, glutamate dehydrogenase. In glycoprotein enzymes, differences in carbohydrate content may be responsible for isoenzymes, like for example, alkaline phosphatase. Let's know a few examples of isoenzymes. Lactate dehydrogenase has 5 distinct isoenzymes, including LDH1, LDH2, LDH3, LDH4, and LDH5. Creatine phosphokinase has 3 types of isoenzymes, CPK1, CPK2, and CPK3. Alkaline phosphatase has more than 5 types of isoenzymes, including alpha-1 ALP, alpha-2 heat level ALP, alpha-2 heat stable ALP, pre-beta ALP, gamma ALP, etc. Alcohol dehydrogenase has 5 types of isoenzymes, class 1, class 2, class 3, class 4, and class 5. Now, I'm going to discuss about these 4 enzymes and their isoenzymes one by one. First of all, lactate dehydrogenase and their isoenzymes. Lactate dehydrogenase is the most thoroughly investigated isoenzyme. Systemic name is L-lactate NAD plus oxidoreductase. EC number is 1.1.1.27. It catalyzes the interconversion of lactate and pyruvate. That means in this reaction, lactic acid is oxidized to form pyruvic acid. Simultaneously, NAD plus is reduced to form NADH. There are 5 isoenzymes of lactate dehydrogenase: LDH1, LDH2, LDH3, LDH4, and LDH5. LDH is tetrameric enzyme, that means it is made up of 4 polypeptide subunits. There are 2 types of subunits of LDH: H, that means for heart, and M, that means for muscle. These 2 subunits are produced by different genes. H subunit is acidic in nature, M subunit is basic in nature. There are 5 subunits of LDH: LDH1, LDH2, LDH3, LDH4, and LDH5. The isoenzymes contain either one or both the subunits, giving LDH1 to LDH5. Like LDH1 means 4 H subunits, LDH2 means 3 H subunit and 1 M subunit. LDH3 means 2 H subunits and 2 M subunits. LDH4 means 1/8 subunit, 3 M subunits, and for LDH5 that means 4 M subunits. Principal tissue of origins for LDH1 and 2 are heart and RBC, whereas for LDH3 it is brain and kidney. For LDH4 and 5 it is liver and skeletal muscle. 5 isoenzymes of LDH can be separated by electrophoresis, either by glucose or star gel or agarogel electrophoresis, as they have different electrophoretic mobility. LDH1 has more positive charge and fastest in electrophoretic mobility towards the anode, while LDH5 is the slowest moving. From these two figures, it is clear that LDH1 has more positive charge, therefore it is fastest in electrophoretic mobility, whereas LDH5 is the slowest moving. LDH1 and LDH2 cannot be destroyed by heat at 60 degrees Celsius. LDH3 can be destroyed by heat at 60 degrees Celsius but partially. But LDH4 and LDH5 can be destroyed at 60 degrees Celsius. That means LDH1 and 2 are more heat stable as compared to 3, 4 and 5. LDH1 concentration is 25% in normal serum in human, LDH2 35%, LDH3 27%, LDH4 8% and LDH5 5%. That means concentration of LDH2 is maximum and concentration of LDH5 is minimum in normal serum in human. Let's know the significance of differential catalytic activity of LDH isoenzymes by comparing LDH1 and LDH5. LDH1 is predominantly found in heart muscle and is inhibited by pyruvate, which is the substrate of this enzyme. Whereas LDH5 is mostly present in the skeletal muscle and inhibition of this enzyme by pyruvate is minimal. In case of LDH1, pyruvate is not converted to lactate in cardiac muscle but is converted to acetyl-CoA which enters citric acid cycle. Whereas in case of LDH5, that means in skeletal muscle, pyruvate is converted to lactate. Pyruvate is not entering into the TCA cycle. LDH1 has high Km, that means low affinity for substrate, whereas LDH5 has low Km, that means high affinity for the substrate, which is pyruvate. The differential catalytic activities of LDH1 and LDH5 in heart and skeletal muscle respectively are well suited for the aerobic, that means the presence of oxygen, and anaerobic, that means absence of oxygen conditions prevailing in these tissues. Isoenzymes of lactate dehydrogenase have immense value in the diagnosis of mainly heart and liver-related disorders. If you see the electrophoresis data of 5 isoenzymes of LDH, you can find that in case of healthy individuals, the activity of LDH2 is higher as compared to LDH1 and other isoenzymes present in the serum. Whereas in case of liver diseases, that means in case of acute hepatocellular injury, hepatitis, passive congestion, and active cirrhosis, increased activity of LDH5 is observed in the serum. In case of myocardial infarction, LDH1 is much greater than LDH2 and this happens within 12 to 24 hours after infarction. In case of lung diseases, that means in case of pulmonary hypoxia, extensive pneumonia, pulmonary congestion, viral pulmonary infection, And in case of cardiac failure, multiple myeloma, and lymphoproliferative disorder, increased activity of LDH2 and LDH3 is observed in the serum. Assays of LDH can measure the amount of LDH present in the serum that leaks from the tissues when damaged. It involves this simple reaction where L-lactate and NAD plus react to give pyruvic acid, NADH and H plus. This is a reversible reaction. Therefore, the conversion of pyruvic to lactate or reverse reaction of oxidation of lactate to pyruvate can be monitored spectrophotometrically. It is more convenient to assay LDH activity in the direction of L-lactate oxidation. That means lactic acid to pyruvic acid conversion, because NADH is produced during this reaction process, and NADH absorbs light at 340 nanometer and NAD plus does not absorb light at 340 nanometer. The reaction thus can be measured as an increase in absorbance at 340 nanometer. Clinical diagnostic laboratories assess the rate of production of NADH that changes the optical density of sample measured spectrophotometrically at 340 nanometer. Typically, the normal range of LDH is between 140 to 280 unit per liter. However, the clinical interpretation depends upon the signs and symptoms of the patient. LDH activity in the RBC is 80 to 100 times more than that in the serum. Hence, for estimation of LDH or its isoenzymes, serum should be totally free from hemolysis, or else false positive results will be obtained. The serum usually has a higher level of LDH as compared to plasma because of LDH release during clotting. The LDH activity also increases during strenuous exercise to generate lactic acid under normal physiological conditions. Now I am going to discuss about the creatine phosphokinase and their isoenzymes. Creatine kinase or creatine phosphokinase catalyzes the interconversion of phosphocreatine to creatine. CPK exists as three isoenzymes: CPK1, CPK2 and CPK3. Each isoenzyme is a dimer composed of two subunits M and B or both. Like in case of CPK1, two B subunits are present. In case of CPK2, one M subunit and another B subunit is these two subunits are present. In case of CPK3, two M subunits are present. CPK1 is present in brain, CPK2 is present in heart, CPK3 is present in skeletal muscle. Let's know the diagnostic importance of CPK. In healthy individuals, the isoenzyme CPK2 is almost undetectable in serum with less than 2% of total CPK. Whereas after the myocardial infarction, within the first 6 to 18 hours, CPK2 increases in the serum to as high as 20% against 2% normal in healthy individuals. CPK2 isoenzyme is not elevated in skeletal muscle disorders. Therefore, estimation of the enzyme CPK2 is the earliest reliable indication of myocardial infarction. After the myocardial infarction, within the first 6 to 18 hours, CPK2 increases in the serum, whereas LDH rises within 12 to 24 hours after infarction, peaks in 2 to 3 days, and gradually disappears in 5 to 14 days. Let's know about alkaline phosphatase and their isoenzymes. Alkaline phosphatases are a group of isoenzymes located on the outer layer of the cell membrane. They catalyze the hydrolysis of organic phosphate esters present in the extracellular space. They are hydrolyses responsible for removing phosphate groups from many types of molecules, including nucleotides, proteins, and alkaloids. The process of removing the phosphate group is called dephosphorylation. It is called alkaline because it functions best at pH 9. Zinc and magnesium are important cofactors of this enzyme. Although alkaline phosphatases are present in different body tissues and have different physiochemical properties, they are true isoenzymes because they catalyze the same reaction. The majority of alkaline phosphatase in serum is released from the liver and bones and in small amounts from the intestine. In the liver, alkaline phosphatase is cytosolic and present in the canalicular membrane of the hepatocyte. Alkaline phosphatase is present in decreasing concentrations in the placenta, ileal mucosa, and kidney. Serum ALP is of interest in the diagnosis of two main groups of conditions: hepatobiliary disease, that means liver disease or damage to the liver, and bone disease associated with increased osteoblastic activity. Normal ALP levels in adults are approximately 20 to 140 IU per liter, though levels are significantly higher in children and pregnant women. Some conditions which cause large amounts of ALP in the blood include rapid bone growth during puberty, bone disease such as Paget's disease or cancer that has spread to the bones, a disease that affects how much calcium is in the blood, vitamin D deficiency, or damaged liver cells. Serum ALP levels are age and gender dependent. Levels are high during childhood and puberty due to bone growth and development. The decrease in level in the 15 to 50 year age group is slightly higher in men as compared to women. These levels rise again in old age. As many as 6 isoenzymes of alkaline phosphatase have been identified. ALP is a monomer. The isoenzymes are due to the difference in the carbohydrate content, that means presence of sialic acid residues. The most important ALP isoenzymes are alpha 1 ALP, alpha 2 heat labile ALP, alpha 2 heat stable ALP, pre beta ALP, gamma ALP, etc. Increase in alpha-2 heat level ALP suggests hepatitis, whereas increase in pre-beta ALP indicates bone diseases. Alcohol dehydrogenase is also known as hangover enzyme. Let's know about this enzyme and their isoenzymes. Alcohol dehydrogenase or ADH and mitochondrial aldehyde dehydrogenase or ALDH2 are responsible for metabolizing the bulk of ethanol consumed as part of the diet, and their activities contribute to the rate of ethanol elimination from the blood. ADH catalyzes conversion of alcohol to aldehyde. They are expressed at highest level in liver but at lower levels in many tissues. This pathway probably evolved as a detoxification mechanism for environmental alcohols. However, with the consumption of large amounts of ethanol, the oxidation of ethanol can become a major energy source and particularly in the liver interferes with the metabolism of other nutrients. Alcohol dehydrogenase or ADH is a zinc metalloprotein with 5 classes of isoenzymes including class 1, 2, 3, 4 and 5. They arise from the association of 8 different subunits including alpha, beta, gamma, pi, delta and mu. These 5 classes of ADH are products of 5 gene loci from ADH1 to ADH5. Location of ADH1 is liver, location of ADH2 is liver and lung, location of ADH3 is liver and stomach, location of ADH4 is liver and cornea, ADH5 is present in most tissues, ADH7 is present in stomach, esophagus and other mucosa, ADH6 is present in liver and stomach. Class 1 isoenzymes generally require a low concentration of ethanol to achieve maximal activity. That means Km value is low. Whereas class 2 isoenzymes have a relatively high Km value. Class 3 ADH has a low affinity for ethanol and does not participate in the oxidation of ethanol in the liver.
Thank you very much for watching this video. Kindly like, share and subscribe this channel. Don't forget to press the bell icon in order to get the notification.